Okay, before I was cut off on the last part, this is where we ended up. So we looked at these two different types of ring structures that can form with two different types of carbohydrates. So here's ribose and fructose, and we talked about that. The idea is that in aqueous solutions in the body, which we are basically a watery environment, these linear chains of carbohydrates can cyclize and form these cyclic structures that you see here. They aren't truly aromatic, so when we use the term aromatic, we're really looking at hydrocarbon groups, but I just wanted to point out that it has that kind of a appearance, right, where we have a cyclic structure. We don't use the term cyclo in front of these at all because these are sugars. So we'll keep going here. All right. She says to the next one. There we go. Okay, we got it. So these are disaccharides, which are formed when two monosaccharides are linked together. And as they link together, we kick out that water molecule. So you can see the water, the part that becomes the water molecule is highlighted in red on these two individual monosaccharides above glucose and fructose. And when they combine by dehydration synthesis, they are forming a covalent bond, and that bond has a special name. So if we're talking about carbohydrates, these monomer units joining larger and forming larger structures, the bond that forms between this monosaccharide and this monosaccharide is called a glycosidic bond. And glycosidic, when you think glyco, think sugar, because in general that's sort of the prefix where we would see in a lot of sugar words that refer to sugar. Water's released. We have these linkages that happen between particular carbons here. So I'll talk about counting carbons later, but in this kind of a structure where it has this sort of pentagon structure in fructose, we start by numbering the carbons in one spot. So if we said that this is carbon 1, this is carbon 2, 3, 4, 5, and then the sixth one is down below that plane of the ring. And on this 6 carbon structure of glucose, because glucose is here, we see that we start numbering the first carbon right here. So here's carbon 1, 2, 3, 4, 5, and then 6 is up here above the plane of the ring. So we can say that this is a glycosidic bond, and we can also call this linkage, which is just linking together, a 1 from the first carbon on this one to the second carbon over here. So a 1 to 2 glycosidic bond or a glycosidic linkage. Here's some other common disaccharides that we can see a lot. So maltose grain sugar, so something you would find typically in something like bread. Lactose, you've heard of that one, that's milk sugar. And then sucrose, which is table sugar, which is the one we just looked at. All of these are made through the same process, dehydration synthesis. Water gets kicked out, forming this glycosidic linkage. And if we want to be specific, we can say which of the carbons are linked. And we can even say not only which way are they linked, but are they linked in this sort of downward position or are they linked in the upward position? But we'll talk about that a little bit soon. Polysaccharides are, as you guess from the word, they have lots of these. They're long chains of these monosaccharide units that are joined by dehydration synthesis, creating a bunch of glycosidic linkages. Some of them can be just long chains. They can be straight chains, or they can get branch structures. We're going to see where they're kind of important. You don't need to know their molecular weight, but it's just to say that they can be big. So let's look at some examples. Common example of polysaccharide is starch. And starch is composed of two types of units in this, or monosaccharide. So there's an amylose and something called amylopectin. and these are joined in a very particular way. So this is the amylose one up here. So looking at these individual units, we see basically these are all glucose monomers that are joining, in this case, in a kind of this straight chain. So that's really the amylose version. And then there's, and the way they're joined, so you can see over here this idea of alpha 1 to 4 glycosidic bonds, or alpha 1 to 6 glycosidic bonds. Those are ways that these can join. And if it's the amylose one, the one that's at the top here, that is forming by a alpha, meaning it's at the direction that we see this molecule joining with the other one. Don't worry about that yet. I will talk about that in class. It's a alpha 1 to 4 glycosidic bond. And then notice in this structure above, you have the brackets around the middle one, and that's because it's saying that this gets repeated over and over and over and over. And then the one below, amylopectin, is a branched type of structure. So you can see here we have a little similar one to the one above. You have this little chain with repeated units, and then notice that it forms this branching down below, which when it branches, we have this joining this particular bond, this glycosidic bond between this one here and the one below it is a 1 to 6 bond. So the first carbon on one of the ring structures to the 6th carbon on the other. Now, the real importance of some of that is the way in which those things happen, the way in which they form, will make a difference as to whether or not it's something that we can digest or something we can't, or structurally what is it used for. So when we see these unbranched chains that are instead not called alpha bonds, but instead are called beta 1 to 4 glycosidic linkages. In other words, we have the carbon 1 on one side and carbon 4 on the other are joining. And so if you look at it, it's really hard to tell, but if you look at it, every other one is flipped upside down. So the one good marker of it is see how this CH2OH is down here on the first one, and then here it is on the second, and then here it is on the third. So each of these units is flipped every other one, which causes this 1 to 4. That's called a beta glycosidic linkage. Now, what that does is it creates that stuff we call fiber. So these are fibers. This is what it would result in which build the walls of plants. All the plant cells are going to have this kind of arrangement. And that's all good for plants, especially because it's very strong. But what it isn't good for us is we can't digest it. We don't have enzymes to break these beta 1 to 4 glycosidic linkages. Where we do have enzymes to break these alpha 1 to 4 or alpha 1 to 6 glycosidic linkages. Let me clarify here, though. Starch has both of these. This is what I'm trying. So this picture is, I was kind of taking them individually, but something you call starch, or things which are starchy, are going to have both of these combined usually. Cellulose only has one type, and they're all nice and straight and linear and fibery that we can't digest, so it goes through our system as fiber. The next topic is lipids, and I'm going to save this for our next installment of my mini tutorial videos. So that's it on carbs, and hope that was helpful.